So, good afternoon. So, this one. So, today we will start with the inhibitions, different types of inhibitions, and how actually these inhibitions affects the clinical enzymology, how it will be used for the treatment, the different drugs, how they act and their different roles. So, enzymes, if you see the activity of the enzymes as we have moved forward, inhibition is enzyme inhibition and enzyme action is one of the important aspect. So, when we see these things, we see there are different types of models for the inhibitions or different theories behind it. Competitive and non-competitive, allosteric, multiple forms of the enzymes, which we will again discuss in details in the isoenzymes. What are isoenzymes, their class forms and the uses. Then you have the reversible covalent modification, proteolytic activation and its importance, and controlling the amount of the enzyme that is present. So, controlling the amount of enzyme that is present in the particular setup actually will give you an idea how the enzyme substrate reacts in the presence of enzyme and catalyst to give the products. So, inhibitor molecules are competing with the normal substrate molecules for binding with the active site of the enzyme generally, because inhibitor you see is a structural analog of the substrate. So, since inhibition, what we see in the enzymes, because the inhibitor is a structural analog of the substrate, enzyme plus substrate actually gives the enzyme substrate, and enzyme substrate will ultimately it will go for the giving the products. So, enzyme plus substrate reacts in a particular presence of a catalyst or a non-catalyst with some activators or modulators to give the product. Since enzyme inhibitor complex can only be used to reform the enzyme and inhibitor, the number of enzyme molecules available for the enzyme substrate formation is reduced. So, suppose there are 100 molecules of the enzyme and 100 molecules of inhibitor is being competing. So, what will happen is that half of the enzyme molecules will be trapped by the inhibitor and only other half will be available for the catalysis. So, 50% will be available for the reactions and 50% will go for the catalysis part. So, this is how actually the enzyme substrate react in presence of inhibitor and the absence of inhibitor. Since the effective concentration of the enzyme is reduced, there will be the reaction velocity is also decreased. Effective volume reduced means the velocity of the reaction decrease, because slowly they are using up the molecule which is actually coming with the substrate and the product formation ultimately. So, this is one example. What you can see is that malonate inhibits the succinate dehydrogenase enzyme. So, succinate dehydrogenase is one of the enzyme, important enzyme, while malonate, when it comes to the red two parts on the thing, means it is an inhibitor. So, what conversion takes place? Here comes the role of the flavin adenine dinucleotide, the coenzyme which forms the flavin adenine dinucleotide hydrate, that is H2, FADH2 that will be coming to. So, here the conversion takes place is from succinate to the fumarate. So, this is actually how actually the inhibitor plays a role, how actually the enzyme to substrate is taking place, and what actually what the thing you should be noting down in the clinical use. So, in a competitive inhibition, now you come to the different types of inhibition. You have heard already before also, or you have studied that what there are different types of inhibition like competitive, uncompetitive, non-competitive, allosteric, suicide, feedback. So, all slowly these will be coming one by one for your understanding and how it is being actually clinically used or relevance. So, in a competitive inhibition, the inhibitor will be a structural analog of the substrate. Structural analog means the inhibitor will be same as that type of the substrate. It is an analog means something like the substrate will be there. So, competitive inhibition is usually reversible, or excess substrate actually abolish the inhibition. Excess substrate means substrate to product formation, substrate will need to react with the enzyme. So, it is an optimum amount of that, so that they react together in the presence of enzyme to form the product. So, they needs this optimal thing, and the excess substrate if it is there, it will abolish or stop the inhibition process. In the case of competitive inhibition, the Km is increased in presence of competitive inhibitor. Definitely the Km would be increased. Km increased, the affinity will be, yes. So, the relationship between the affinity, the Km and the velocity is very important throughout the enzyme kinetics or enzymatic reactions. Thus, competitive inhibitor apparently increases the Km. In other words, the affinity of the enzyme to a substrate is apparently decreased in the presence of a inhibitor, but Vmax will never be changed. The Michaelis-Menten constant or the other things, the Km value will be depending on the Vmax, but Vmax will not be changed. So, this type of inhibition, apparently role of inhibitor can be therapeutically exploited in different disease, where competitive inhibition is there. We will come to the different examples where many drugs competitively inhibit a particular pathway or particular enzymes and it is being successfully used in the clinical regimen or the clinical aspect. So, these already you have known, that is substrate saturation curve in presence and absence of competitive inhibitor. The first one, A is the Lindbergh plot and B is the Michaelis-Menten saturation curve. So, Lindbergh plot we have discussed when I have discussed the Michaelis-Menten in the last class, that it is called the double reciprocal plot. So, you have to move from not from the 0 0 plus plus quadrant, you move from the plus minus quadrant and other quadrant to check actually how actually the enzyme moves or how actually the enzyme affects or how it actually reacts in the presence of the substratal thing. So, this is the importance of the Lindbergh plot and the Michaelis-Menten equation of the. So, clinically importance, what are the different importance of competitive inhibition? So, first importance of the competitive inhibition is the different sulfonamides that we are taking. You have to know why are you actually taking this on inhibition, why are you studying, what is the clinical relevance, how actually it will be helping us in the clinical setup or diagnosis is very important for us. So, what happens in the sulfonamides? They are commonly employed antibacterial agents. Bacteria synthesize folic acid by combining PABA with the pteroyl glutamic acid. Sulfur drugs, its success actually is a structural analog of the PABA, that is full form is para-amino butyric acid, which inhibits the folic acid synthesis in bacteria and they die. So, how actually the sulfonamides through the reaction with the PABA and the different role of the different bacteria and the other things in presence of the folic, which actually inhibits the folic acid. Folic acid, you know, it will be coming, it has a different roles, so functional property. So, they can actually act in the competitive inhibition pathway. Next drug is the methotrexate. Methotrexate is a structural analog of the folic acid. Methotrexate, what you see as the drug, is a structural analog of the folic acid. That means, it is structurally similar to that of the folic acid, so that they can completely inhibit, that is competitively inhibition means it will be acting together with the methotrexate and the folate will be, and so they can inhibit the folate reductase enzyme. So, something looking like a similar acting or as that of the similar like folate, if you take in the case of methotrexate, it is an analog of folic acid. So, analog of folic acid means it will compete with the folic acid. So, it will inhibit the, if methotrexate is given, it will inhibit the enzyme, that is folate reductase, and methotrexate will be acting in the pathway. Where it will be important? It will be in the cancer diagnosis or cancer detection, that is it is a cancer drug. So, it is prevention of cancer and other things, this methotrexate will be useful, and since it is known as a anti-cancer drugs. So, this is here example of another example of competitive inhibition. Next comes dicumarol. It is a structural similar to that of vitamin K. A, D, E and K comes under the fat soluble vitamins. So, vitamin K falls under the category of fat soluble vitamins and can act as an anticoagulant by completely inhibiting the action of the vitamin K activity. So, you see different examples which I am showing over here has different action, but all are clinically very important for particular disease or condition. Similarly, we have the INH or isonicotinic acid hydrazide, which is an anti-tuberculous drug. It is structurally similar to pyridoxal or pyridoxal, and prolonged use of INH or isoniazide may cause pyridoxal deficiency and peripheral neuropathy. Pyridoxal deficiency, pyridoxal means it comes from pyridoxine. So, from there the pyridoxal came and it maybe goes for the peripheral neuropathy. So, you see if one drug is being used to treat a particular disease, there are chance that it may lead up to some other clinical conditions. So, what is competitive inhibition? Examples of competitive inhibition you should know with examples. Examples means with particular cases. which drug, where it acts, how it functions, and with the function of the competitive inhibition which is reversible nature, are there any side effects or any other aspects that to be taken care of. Understood? Any doubt you can ask. So, these are the clinically useful competitive inhibitors. You want the previous slide? Write it down. So, just 4 important things which are very important and you must know I have given. You can take other examples also, but whenever you give examples or whenever you write answers about the competitive inhibition other things, you have to give these examples and you have to check and show the clinical relevance in which disease or which condition they are active. Okay. That you should not forget. Peripheral neuropathy, nervous system problem, neuropathy, nerve related problems.
Sir, how folate reductase enzyme causes anti-cancer?
Folate reductase is just an enzyme pathway, it is a path of the cancer activity. When you see in the thing cancer pathway, there is one enzyme that is being targeted. Methotrexate is a known anti-cancer drugs like Taxol, doxorubicin, it is known. They target, see any drug there should be a particular target. We are targeting that folate reductase in the cancer pathway for this activity which is methotrexate, and this inhibition is competitive in nature which is reversible. Okay. So, these are clinically important useful competitive inhibitors. You should actually note down in the tabular form, it is easy for you to remember. We have the drug, you have the enzyme inhibited, that is how what is the enzyme that is there, then you have the clinical use. Allopurinol is the drug, enzyme that is inhibition is xanthine oxidase, clinical use is gout. Now, why gout and xanthine oxidase? Because in the mechanism of gout, when gout happens in that pathway, xanthine oxidase is an important enzyme that is in the pathway, that is being actually targeted by allopurinol to stop the gout. Gout occurs due to the excess accumulation of the uric acid in the joints and the thing, which may causes pains in the joints and form the tophases or toffee. Dicumarol, it is a vitamin K epoxide reductase, it acts as an anticoagulant. Anticoagulant is required when blood clotting problems are there, so we need the anticoagulant to make it in a proper function. Penicillin, transpeptidase, antibacterial. Sulfonamide, steroid synthesis, antibacterial. Trimethoprim, FH2 reductase, that is antibacterial. Pyrimethamine, FH2 reductase, that is antimalarial. Methotrexate, folate hydrogen reductase, FH2, that is reductase, anti-cancer. 6-mercaptopurine, that is enzyme is adenylosuccinate synthetase, it is anti-cancer. 5-fluorouracil, thymidylate synthase, that is anti-cancer. So, see in anti-cancer category and antibacterial category, it is not that it is acting on the same enzyme, it may act on a different enzymes, but mechanism of action and understanding will be the same, that it will have this anti-tumor or anti-cancer property, antibacterial property and particular disease. So, in anti-cancer you see 3 are there: folate reductase, adenylosuccinate synthetase, thymidylate synthase. So, understood competitive inhibition, you should know definition, what are the properties of competitive inhibition, what is its role in reversible in nature, how it functions and examples. Examples with the clinically important competitive inhibitors which are known drugs, which pathway they act through which enzyme and their clinical uses. Understood? Okay. Because we have to move one by one to the other things. Next is azathioprine is there, phosphoribosyl amidotransferase is anti-cancer, cytosine arabinocyte, that is DNA polymerase anti-cancer drug, acylsobit, that is DNAP of the virus, that is antiviral, neostigmine acetylcholinesterase is myasthenia gravis, one disease of the neuropathic nature, neural disease, that is myasthenia gravis is there. Alpha methyl dopa is dopa carboxylase, decarboxylase, that is hypertension, alpha dopa is being there. Lovastatin, cholesterol lowering or anti-cholesterol or hypercholesterol drug is there, lovastatin. It acts on the hydroxymethyl butyryl coenzyme reductase, which is the rate limiting step of the cholesterol metabolism. Then you have the Tamiflu or the neuraminidase which actually cures the influenza. So, you see so many drugs, the mechanism of the drugs which actually acts in different ways for the different diseases conditions for curing particular types which acts by the principle of the competitive inhibition. Then comes non-competitive inhibition, that is irreversible in nature. I told you competitive inhibition is reversible. They compete with the structural analog of the particular substrate, so that they go in the competition and affects goes and reacts with the enzyme competitively to give the product or the formation of the thing or to stop something like that. Whereas, when you come to the non-competitive part, that total thing will be irreversible in nature. ठीक है, तो एक पाथवे में अगर चला गया तो वी कैन नॉट बी कमिंग बैक टू द ओरिजिनल पोजीशन अदर थिंग्स. So, this is the main difference between the competitive and non-competitive in nature. There is no competition between substrate and inhibitor. The inhibitor usually binds to a different domain on the enzyme other than the substrate binding site. This is very important. Substrate binding site छोड़ के दूसरा जो domain होगा, it will bind in that particular domain, so that there is no competition at all, but it will bind at a particular place. Since these inhibitors have no structural resemblance to the substrate, an increase in the substrate concentration generally does not relieve this inhibition. Since this is important, which is written in the board, since it has no structural resemblance means similarity, to that of the substrate, an increase in the substrate concentration generally does not relieve this inhibition. Substrate concentration, that this thing, the inhibition, there is no direct any relation, because it does not compete with the substrate at all. Examples are cyanide, which inhibits the enzyme cytochrome oxidase, very important. Fluoride will remove magnesium and manganese ions, and so will inhibit the enzyme enolase, and so consequently the pathway, the glycolytic pathway. Iodoacetate will inhibit the enzymes that having the thiol group SH in their active centers. British anti-leucite or dicumarol, it is used as an antidote for heavy metal poisoning. So, these all the four examples I have given is of non-competitive inhibition, that is irreversible in nature, and they are clinically important. So, we are going just the basic structural aspect we should know, and more further reading and other things you can get the concept of the things when you come to the discussion of the different clinical cases and clinical scenarios, which will give you a better understanding of the case things. Like cyanide poisoning, a person comes with this problem, cytochrome inhibition, he has these type of features, this drug is given, then what type of inhibition comes? You can, that comes in a questionnaire, that is the objective structured clinical examination, practical examination, structural questions, these clinical aspects of the different views, case studies, these things we will be discussing. So, this if you look in a nutshell or a schematic view, this is what happens. You have the enzyme, you have the substrate, but you see the substrate and the product binding and the non-competitive inhibitor is completely different. So, this diagram you should remember, and you just draw the basic things to understand how actually this is related to the substrate concentration, where the substrate lies, where the enzyme lies, how the product is formed, and what is the role of the non-competitive inhibitor. Where non-competitive inhibitor is given in blue in color and the substrate is given in red in color, and the black color is the enzyme and the product will be given over there. So, inactive enzyme will be there, substrate will be moving out and no product is formed. So, it is stopping at a particular level, not leading to the formation of the product. Once the product is not formed, means there is no fear of any damage or any hazards which will be caused due to the thing, or any disease condition propagating or continuing of the thing. So, this diagram you should draw. This is important. How actually this non-competitive inhibitor will be there, how the substrate will be reacting, it is coming out of the thing. So, it is not reacting, inactive enzyme, the enzyme it is not functional over here or inactive in nature, and ultimately we are not having any product, and this total path thing is irreversible in nature. That is most important. Next, with inhibitor and without inhibitor, what happens? So, saturation curve will be for non-competitive inhibition is different from that of competitive. These two graphs or is very important when you write or answer competitive inhibition and non-competitive inhibition. You have to give whenever you write answers to these type of things. First is definition, characteristics property, examples, and the graph. Graph actually gives you an understanding at what happens to the enzyme kinetics of the reaction with and without inhibitor. So, here you can see two types of thing. One, you see the Lindbergh plot, and B, we see the Michaelis-Menten saturation curve. The velocity is reduced, but Km, that is the Km is the Km remains the same, not changed, but the name what is Km? Yes, of the V max. What he tell as the Km? Other name of Km? Michaelis-Menten constant. Michaelis-Menten constant. That is important. Other name of Km, because they can give you both the ways. Importance of Km and other functional aspect. So, now comes the most important comparison between the two types of inhibition, and this comparison should be on different types. Whenever you study biochemistry, it should be different segments. Which are you comparing? Where are you comparing? What are the sites or the parameters you are based on which you are doing the comparison? So, first is acting on. Competitive inhibition is active site, non-competitive may or may not. Structural of the inhibitor is substrate analog, unrelated molecule. Inhibition is generally reversible, competitive, generally non-competitive is irreversible. Excess substrate inhibition is relieved, but non-competitive no effect, because it does not go for the any comparison or nearby to the substrate. Km is increased, non-competitive not change. Vmax no change, in non-competitive decrease. Significance drug action maximum, non-competitive more of a toxicological effect, because cyanide poisoning and other things most come with the toxicology effect. Heavy metal poisoning, cyanide poisoning, more of a toxicological part that comes as a non-competitive, whereas for drug therapy, therapeutics, competitive. This table you should write down properly. This is the key thing which we have discussed till 10-15 minutes now. Understood? Simple. Go through the things. So, the basic structure of these things you should know, as I am taking in the class. Extra things you can add, but for the way it is presented, that you should know. Then only can you answer these things in the, it will be easy for you to answer in the any exams or for your other assessments. When you write in the exam also, you should follow this tabular form. So, it will be easy for you to understand and write it. And one more extra thing you can add, the examples. Examples of competitive inhibitor, or the any example where competitive inhibition is utilized, and any example where non-competitive inhibition is being utilized or used. So, one is toxicological, one is more of a drug action. Understood?
Yes, sir.
Now comes uncompetitive inhibition. पूरा लिखा कि नहीं लिखा? लिख लिया ना? Okay. Next I can change. So, uncompetitive inhibition, from the term or the title you can understand, there is no competition at all. Uncompetitive. कंपैरिजन ही नहीं है, दोनों में कुछ कंपैरिजन ही नहीं करना चाहिए. So inhibitor doesn't have any affinity for free enzyme. Inhibitor binds to the enzyme substrate complex, but not to the free enzyme. In such cases, both Vmax and Km are decreased. Inhibition of, like example is that of placental alkaline phosphatase, it is a coenzyme, ALP coenzyme, it is also called the Regan's isoenzyme. By phenylalanine is an example of uncompetitive inhibition. Alkaline phosphatase have different forms of enzyme, it is an isoenzyme. So, one of the thing is the Regan's isoenzyme, it is being inhibited by phenylalanine. You know phenylalanine already, in the amino acid class it is being already taken phenylalanine, it comes on the aromatic amino acid category and it is an example of uncompetitive inhibition. So, if you see the graph 1 by S with inhibitor and without inhibitor, you get like a, in the negative quadrant you have the minus 1 by Km and minus. So, with inhibitor what will happen and without inhibitor. So, this parallel line will be coming, but there will be no competition between the two things at all. No, Km straight रहेगा, Km should be Vmax decrease both.
So we can see from the graph that the Km intercept is more
Generally नहीं करता इस पे, intercept नहीं करता generally, but if you see the proportionately, it is with inhibitor, it is more taking more, but you have to calculate and check it with the thing generally, general aspect में. Next comes the suicide inhibition. It is also known as the mechanism based inactivation. It is also known as the mechanism based inhibition. The inhibitor makes use of the enzyme's own reaction mechanism to inactivate it. In suicide inhibition, the structure analog is converted to a more effective inhibitor with the help of enzyme to be inhibited. Example is ornithine decarboxylase, ODC, that converts the ornithine to putrescine, which is necessary for the polyamine synthesis or polyamines, synthesis of the polyamines. When the ODC in tryposinoma is inhibited, multiplication of the parasite is being arrested over there. So, how actually ornithine decarboxylase enzyme reacts, what is its role and what are the inhibitors. The inhibitors of ODC enzymes such as DFMO or difluoromethyl-ornithine, it is an inhibitor, has been effective against this sleeping sickness or trypanososis. So, sleeping sickness, this is one drug that is being used and this is being one of the example of the suicide inhibition. A similar mechanism is observed in case of allopurinol, which is oxide by xanthine oxide to alloxanthine, that is a strong inhibitor of xanthine oxidase. Again allopurinol you have studied in the which enzyme, which inhibition I have showed you? Competitive inhibition. There comes the confusion. Allopurinol is used actually a competitive inhibitor or a suicide inhibitor. This is a big thing. Now you tell me what is your judgment about this. Because allopurinol is competitive in the gout. Here also it is going and showing as the both the example is there. Then when you write in the exam, what should you write? Many of the books, many of the things it is going for things that how actually allopurinol acts. Generally the concept is that since allopurinol goes as a structure analog formula, structural analog of the thing, generally it is acted as a competitive, not comes on the suicide thing. The drug, because allopurinol will be used for curing the gout. Gout means which enzyme it will be targeting? Xanthine oxidase. So, xanthine oxidase will be there, means it needs some structural analog and thing similarity, and it goes in that way. But many places and many books this is being also highlighted in the suicide inhibition.
in the top of the slide it says that in suicide inhibition there is a structural analog which is converted to an effective inhibitor.
Yes.
So can we say that if an inhibitor does not get converted into some other compound, then it is competitive inhibition, and if it gets converted it is suicide inhibition?
It is right in that way, but it is taken into both the account because it is thinking that this analog, see similar mechanism they are thinking that that may not happen in the case of allopurinol based, which is given in the competitive inhibition. That is why what I told is that it seems though it is given in the suicide inhibition, but classically speaking or thinking in the structural analog part, it should come in competitive inhibition, not in the suicide inhibition. But many of the books is, that is why I have written over there also. You have seen the books, it will be written like that. it should not come directly over there, it should go for the competitive inhibition only.
Sir, it can be both, right?
It can be both, but see, if you go more of a structural analog part, classical example definition part, then it should go in competitive, that because it is competing over there. But if it is changed, the top it is written, no, inhibitor makes the enzyme own reaction to inactive, the structural analysis is converted to more effective inhibitor with the help of enzyme to be inhibited. That line is the, yes, that line is the main key to the all these examples, more effective inhibitor. So, if I want do not want to make a more inhibitor, more competitive inhibitor or more better inhibitor, then comes in the competitive. Try to understand, because these queries and these questions will be throughout till you go for the PG. PG also this question is a will be coming and you have to answer back, and you should be strongly giving the judgment that why you want to say it is a competitive inhibition. Similar in the aspirin also there for the suicidal inhibition. Aspirin is a analgesic. Yes, NSAID. Very good. Next comes the allosteric regulation. Allo means what? For the term allosteric means what you understand. It is an enzyme that only has one catalytic site where the substrate binds and other separate allosteric site where the modifier binds. Allo means other. Allo means always, that is other, you remember. That means a seat is there, one person is already occupying the seat, another person can occupy some space is there in the same line or alignment. Here it is happened like that. So, allosteric and substrate binding sites may or may not be physically adjacent, important. Allosteric site or substrate binding site दोनों साथ साथ नहीं भी हो सकता है. It may not be adjacent, it may be far away from other, but there will be two sites. Allosteric means other, allo means one more or other. The binding of the regulatory molecules can either enhance the activity of enzyme, allosteric activation, or inhibit the activity of enzyme, allosteric inhibition. It depends upon where actually it is being, it is adding, or where actually means oriented. That is important. If it is adjacent, that means there will be steric hindrance, steric coordination, that may help the thing, may not help. If it is away from each other, it may work independently, where steric congestion, conformational problems, conformation analysis will be giving up the better spatial orientation, which will not actually disturb the entropy and the enthalpy of the reactions, and it will be a thermodynamically feasible reaction to go through. So, this is actually, that is why allosteric regulation is always gone into two categories. One is allosteric activation and one is allosteric inhibition. That subdivision you should go through. The binding of the substrate to one of the subunits of the enzyme may enhance the substrate binding to by other subunits. One substrate may enhance the binding to the other subunits. The key points you should be noting it down. Examples I will give. This is the example, you can understand from here. See clearly the picture, then you will understand what I want to say. So, action. Activity is fixed to the allosteric site. When active site has correct conformation, the substrate is correctly fixed. Figure 2 shows the inhibitor is fixed to allosteric when the active site is deformed and the substrate could not fix. A is the active site, A is the allosteric, S is the substrate, A is the allosteric activator, IN is the inhibitor, ALN is the allosteric inhibitor. So, activator and inhibitor, that is the most important in this allosteric activation. These diagrams always you should understand with the diagrams for enzyme inhibition, otherwise very difficult. Competitive inhibition diagram I have given you, take the drug and try to understand that. Suicide inhibition also you should go with the diagram, then you will understand that if it is being modulated in such a way that is better than the thing and acts as an inhibitor, okay. Uncompetitive, there is no relation at all. Non-competitive, competitive, suicide, feedback I will come later for the. So, with these diagrams you should draw to understand the better way, and try to give different colors with these things, so that you understand what is actually, which is an inhibitor, which is an analog, which is an activator, you will understand. because this pictorial diagram makes you better and gives you a better understanding than what simply writing the things. It creates an impression on the brain. Very simple. See, this is the way you should understand. You can make it with the Harper. If you see the diagrams after this, you will see how Harper has it is given in biochemistry, how the diagrams and the things. They are made a very crevices and complicated thing. So, once you understand the basic, you can go for the advanced understanding. But you have to go through the things, you have to understand, you have to read through the books. All of you are drawn? Complete it, you have to go through the other part. So, you have to understand the basic things, basic diagrams, differences between all the inhibitions, applications, role of Vmax, role of Km, role of the Michaelis-Menten equation and role of the Lindweber-Buck plot, because that is more used in the laboratory for understanding the enzyme kinetics. So, saturation curve of allosteric inhibition, you agree with me? Positive modulator, sigmoid is a normal curve, negative modulator means it does not make you, it is not helping in the progress of the reaction. Positive, it is moving beyond. Normal is the sigmoid curve, that you know what are the enzyme substrate complex. So, normal is blue, when it is a negative modifier it is in black, and when it is a positive modifier is there it is in red. So, Vmax, half the Vmax you can see the concentration, but x axis is the, we have the substrate concentration, and y axis we have the velocity. Enzyme chapter fully depends upon all the reactions, graphs, examples, nothing more than that, simple forms.
So, is allosteric inhibition the same thing as non-competitive
No,
inhibition?
no,
So, with the definition.
no, no, no. I have given you proper definition. Allosteric means binding than the other side, but in non-competitive it is different, it is irreversible, it is competing in, it is not competing, but other side, but this and that reaction characteristic is different, because if you do not understand the precise classification of the enzyme it will be difficult. Non-competitive is more for all the different type of toxicological mechanisms, whereas allosteric it can be for normal mechanism, oral drugs, or can go for allosteric. There is in allosteric there is advantage is that you have both positive modulation, there is allosteric activation and allosteric inhibition, whereas in non-competitive there is no chance of that. You will find little similarity between the thing, but you cannot club and make things in it.
The site for the analog which is present in non-competitive can't change its shape like
No, it cannot be deformed, no, it is already fixed, here it can be deformed. So, it cannot be changing like that. That is why I have told you, non-competitive is more for toxicological cases, means those cases means that very serious or acute cases it comes, and it is irreversible in nature. Try to understand, irreversible means you cannot go back, like in cytochrome, what happens? Cytochrome oxidase something is there. Cyanide poisoning. Cyanide poisoning case means very difficult to harness it. Heavy metal poisoning. Whereas in allosteric you see it is different. Allosteric you see the inhibitor is not a substrate analog. First of all, inhibitor will not be a substrate analog. It is partially reversible when excess substrate is added. Partially reversible. जो नॉन कॉम्पिटिटिव होता ही नहीं है. That's why I am told when you understand the features, then you will understand what is the difference. Km is usually increased, Vmax is reduced. Effect of allosteric modifier is maximum or near at the substrate constant equivalent to Km, definitely near the Km only this value will be maximum. Most allosteric enzymes processes quaternary structure, they are made up of subunits. Example, aspartate transcarbamylase has 6 subunits and pyruvate kinase has 4 subunits. Mainly this allosteric inhibition will occur in the enzymes which have different subunits. Subunits that will be present when subunits will be present, it is easy to check where actually the binding of the substrate is there, where the analog is there, and how with the binding of the analog with the substrate will enhance the activity of enzyme or inhibit the enzyme activity of enzyme. So, allosteric, always remember there will be most of the allosteric enzymes possess quaternary structure. Quaternary structure of proteins is completed, no, I think primary secondary structure tertiary होगा, quaternary अभी तक नहीं है. तो when you understand, when you see this quaternary structure, you understand what is the bonding, what is the orientation, and how actually it acts. Then only it will give you a proper understanding of this allosteric binding and what are the different examples of these allosteric enzymes. So, now you have to go to the different examples to make you understand. So, what are the different examples of allosteric enzymes? Delta amino-levulinic acid synthase, allosteric inhibitor is heme, aspartate trans carbamylase, allosteric inhibitor, citrulline triphosphate, allosteric activity is ATP, HMG quark reductase or HMG quark means hydroxymethyl butyryl coenzyme A reductase, which is a rate limiting step or rate determining step of cholesterol synthesis. Inhibitor is cholesterol, that is why it is used as a lovastatin and compactin are these type of drugs which inhibits the cholesterol biotin. So, in hypercholesterolemia patient and other things, to minimize or to keep cholesterol under control, this type of enzymes will be activated due to drugs. So, these drugs are given for hypercholesterolemia or yes. Next is phosphofructokinase ATP, that is citrate, AMP and fructose 2,6 diphosphate bisphosphate or fructose 6 phosphate. Pyruvate carboxyl is ADP, allosteric activity acetyl coenzyme A. Acetyl coenzyme A, carboxyl is acetyl coenzyme and citrate is the activator. Other enzymes we have the inhibitor like citrate synthase ATP, carmyl phosphate synthetase 1 is N-acetyl glutamate or NAG, very important. And last is carmyl phosphate synthase 2 is UTP or uridine triphosphate. CPS1, CPS2. CPS1 is in the urea cycle pathway and CPS2 in the which biotin this? Which pathway? Carbamoyl phosphate synthase 2?
Carbamoyl phosphate, it is in urea cycle.
1 to urea cycle, 2 nucleotide metabolism में purine or pyrimidine? Pyrimidine. Uric acid. So, you should know what is the difference between these comes. What is the difference between CPS1 and CPS2? CPS1 और 2 में difference क्या है? Which pathway it acts and what are positive modulator and negative modulator? इसका positive modulator N-acetyl glutamine, an inhibitor, and this thing is the UTP or uridine triphosphate in CPS2. Everyone written down this thing? लिखा नहीं, लिख लो. No talking.
Whether Km will increase or decrease, it will depend on whether it is acting as inhibitor or activator or it is.
पहले पढ़ लो, पढ़ो, तुम पढ़ा ही नहीं पहले. Km doesn't depend upon inhibitor activator. Km will depend upon the how the enzyme substrate reacts and उसके ऊपर depend करेगा. उसके ऊपर depend करके Km का ये रहेगा. You have to read this thing properly. Because always any type of curve it will be half saturation reach, then it will go sigmoidal. ये straight कभी नहीं जाएगा. 50% भी only molecules it will be utilizing, and other thing it goes as thing. That's why in the any enzyme substrate reaction you get a sigmoidal curve. एक time के बाद वो saturate हो जाएगा. Feedback inhibition. The term feedback inhibition means the activity of the enzyme is inhibited by the product of the reaction. Inhibition by hexokinase by glucose 6 phosphate is a very classical, most important, usually widely studied in the glycolytic pathway. So, glucose plus ATP in the presence of glucose 6 phosphate gives ADP and PI. End product inhibition is it is given. Next is carbamyl phosphate plus aspartate transcarbamylase gives carbamyl phosphate plus aspartate plus carbamyl aspartate plus PI. So, what is happened? CTP, the end product will allosterically inhibit the aspartate transcarbamylase. CTP is the end product, that will be, it will be inhibiting the feedback, that means product formation is there, it does not require more substrate to react to give the product. So, from there, from the feedback of the inhibition stops. So, example is one is a glycolytic pathway and the CTP are there. Next is in the ALA synthase pathway, the delta amino acid, this is the first part of the heme synthesis, first step of the heme synthesis. This delta amino levulinic acid is the first thing step which reacts with the glycine in the presence of ALA synthase or delta amino levulinic acid. This ALA synthase gives the delta amino levulinic acid formation. The end product heme will be allosterically inhibit the ALA synthase. See, all these enzymes, there is a feedback or the thing that is being occurring which actually stops reaction of the total process from the substrate to the product. Product formation नहीं करने दे रहा है, उससे इनिबिट होके ये पीछे आ रहा है. So these things will be important for us.
Here the feedback will always be from the end product.
Yes, it is for end product. Feedback, what is mean by feedback? Yes, the final product. Feedback only comes after you have something, no. You give feedback. If you disturb like this, see, things will be very difficult in the class, I am telling you. I have to finish the course, you will only miss. Next week you go for Diwali holidays and other things, you cannot complete. I will continue till 30th, I have no issues. You will be not there, you will miss it. Try to understand. Next is induction. So, these feedback mechanisms always will be depending upon the final product, because feedback you can only give when something is given to feeding, something is given or product provided to you, then you can give the feedback. If I do not take a class and I ask you feedback, nobody can give, and you can give, you people are, you can give the feedback, but that will be biased, you do not know anything, you like something on that. So, always the end product or something which actually we get from the reaction will give you the importance of the feedback. Next is induction. Induction is effected through the process of derepression. The inducer will relieve the repression of the operator site and will remove the block on the biosynthesis of the enzyme molecules. Classical example of induction is the lactose utilizing enzyme in the bacteria. Bacteria में जो lactose utilizing enzymes होते हैं, yes, when the media contains lactose in absence of glucose, that is in, yes, what is that called? You people should, yes, so lac operon concept, that's importance of the induction. That's why I am not given space for you to understand. That's operon concept, lactoperon concept, that is induction, repressor and derepressor theory. That is also comes under the enzymatic reaction. Repression, you can understand. Repressor actually acts as a gene level, because this repressor and induction, the jumping gene theory, the lac operon concept all comes in the molecular biology when you will be there. But basically enzymatically if we see, we can see that it acts as a gene level, effect is noticeable only after a lag period of hours or days, and the number of enzyme molecules is reduced in the presence of repressor molecules. So, if repressor means, what means by repress? That means you are slowing down something, you are making an obstacle for something, you are inhibiting the progress of something, that is the to repress. So, once you repress the enzymatic reactions, always the product molecules will be coming less. So, this is what will happen if you see in the case of a structural gene, that is in the heme, what will be happening. You have a regulator, you have an operator, you have the structural genes. In the regulator you have a aporepressor and a co-repressor. Apo I told other, co means same type. Both called the holorepressor. Like first class I told you what are the difference between apoenzyme, holoenzymes, isoenzymes, coenzymes, cofactors and yes, prosthetic group. So, here also this is a classical example, you should know it. Understood? Next come the covalent modification. the activity of enzyme may be increased or decreased by covalent modification. Covalent modification, till now we have not talked anything of the covalent part. Now we have to come to the covalent modification is also there. It means either addition of a group of enzyme up to the enzyme protein by a covalent bond, or removal of a group by cleaving a covalent bond. Now we come to the covalent bond, which is a strong bond consists of a pi and a sigma bond, which is very difficult to break. It is energy consuming. So, zymogen activation by partial proteolysis is an example of that is covalent activation. Commonest type of covalent modification is the reversible protein phosphorylation. The phosphate group may be attached to serine, threonine or tyrosine residues. When hormone binds to the membrane bound receptor, hormone receptor complex is formed. This enzyme adenylate cyclase is activated. This activation is mediated through G protein. This adenylate cyclase activity, again you can see the thing in the glycogen metabolism pathway. So, this comes under the example of covalent modification. That means, you are using or utilizing the role of the covalent bond, breaking and making of the things in the enzymatic activity. Examples particular where it is happening, you should know. Examples are here. Write down the examples of the covalent modification. So, enzymes, you have the phosphorylated enzyme, that is acetyl CoA carboxyl is inactive, glycogen synthase inactive, pyruvate dehydrogen is inactive, HMG coenzyme reductase inactive will be there, active will be from glycogen phosphorylase, citrate lyase, phosphorylase B kinase, HMG CoA reductase kinase, fructose 2,6 bisphosphatase. So, covalent modification, definition, what are the features, where it acts and examples. All will be mainly of the phosphorylated enzymes, because phosphorylation will be involving the double bond, that concept ATP and on the phosphate will be donated ATP to ADP and PI. When phosphorylation will occur, there will be PO3 minus PO4 phosphate and one bond will be slowly going. So, ATP means ADP plus PI or ATP to AMP plus PPI, pyrophosphate bond will be there. So, those are all high energy bonds. Now comes the absolute specificity. Hydrolysis to urea to ammonia by carbon dioxide is catalyzed urease, it is a specific for urease. This is isovalent, and bond specificity is there. Most of the proteolytic enzymes are showing bond specificity. Trypsin can hydrolyze peptide bonds forming by carboxyl group of arginine or lysine residues in any protein. Absolute specificity and bond specificity. This comes under separate headings, which is being used when the estimation of urea by urease and bond specificity by the carboxyl group of arginine and lysine. You have also the group specificity and stereo specificity. Group means one enzyme can catalyze the same reaction or a group of similar compound like hexokinase can catalyze phosphorylation glucose, galactose and mannose. When I came to the Km value and Vm, I told you affinity. What is the role of hexokinase and glucokinase in different reactions? Stereo specificity means for L amino acids and D carbohydrates. Fumarase will hydrate the fumaric acid to malic, but corresponding cis form malic acid will not be acting around. So, which type of stereo specific? The stereo means structural orientation in the space, specific means for the particular compound. So, lactate deoxygenase act on pyruvate will only on L lactate, but not on the D variety. So, specific L and D almost I hope you know it. What is L and D? Small l and small d, large L and large D, both you should know. Now, two more important things is that body metabolism is controlled by enzyme, very important. This is what is the advance of the part we are discussing. In a pipe, theoretically water flow can flow both ways, but practically the flow is unidirectional. Try to understand. In for something else, so the reaction as a whole tend to go in forward direction only. In the tap, it should be bidirectional the thing, but you see due to the force only one direction you get it. So, what happens is that True equilibrium of chemical reaction in a cell is achieved only when the cell is dead. In other words, living cells avoid the state of chemical equilibrium. Always remember. At the same time, if you analyze a particular metabolite, the concentration is kept constant over a long period, metabolite. This is called a steady state system. For example, blood glucose level is kept within a narrow range because the factors which tend to increase levels are at dynamic equilibrium with the factors that try to decrease the levels. So, we need to keep it at a narrow level for this particular part. Always in the biochemical part we have a range, some are too narrow and some are little bit, but not very broad, because this dynamic equilibrium plays an important role which actually alters the levels. Deficiency of enzyme or activity lead to genetic defects, immoderance of metabolism. Such deficiency may be due to genetic mutations or by infection. Now come drugs metabolism. What is the role of drug metabolism? These are the advanced aspect of enzymatic things. Drugs act as enzyme present in human beings or in pathogen. Penicillin, we have told, it blocks cell wall synthesis in bacteria by irreversible binding of the enzyme transpeptidase. You know already, we have discussed there in the enzyme activity. Many bacteria, however, produce beta lactamase that hydrolyze the penicillin. Another example of the drug is 5-fluorouracil, which is metabolized inside human body into deoxyfluorouridylic acid, which inhibits the thymidylate synthase and which acts as an anti-cancer drug. So, you can understand how actually drug metabolism. Next, this is the most important and the advanced part is what is enzyme engineering part. Enzyme engineering is that enzymes are widely used in food, pharmaceutical and chemical industries, and these bacterial enzymes from time immemorial accomplish fermentation of the different food items. So, yogurt and cheese, what you see and what you see as the fermenting of rice and black gum for the preparing delicious food, I have given you the or examples of this enzyme engineering. More of the food part, we are not going for engineering for the different other things. It is even used for removing stains from the different of clothes also. So, this is called the enzyme engineering, that means using the enzyme in a different aspect of the commercial part. Then you go for the drug designing. Drug designing also you can go, and processive enzymes. Processive enzyme means they continue to act on a particular substrate, but do not dissociate between repetitions of the catalytic event. Examples are DNA polymerase, RNA polymerase, glycogen synthase and fatty acid synthase. These are the advanced part of the enzymes. Multi-enzyme complex, this you should know, that is single enzyme molecule enzymology and the mechanisms. Sequential reactions or single displacement reactions, both substrate combine with the enzyme to form a ternary complex which catalysis forms. Ping pong means ping pong state you have studied in the cell biology part. Here also we have this thing, a group undergoing transfer is first taken from one substrate A, added to enzyme, subsequently the group is taken from the modified enzyme and added to another substrate. Examples are aminotransferases. These are the typical example. Aminotransferase will come in the chapter of the amino acid metabolism part. Reactions where products are formed before all the substrate react are known as the ping pong reactants. Single molecule enzymology is this happens in the recent advance in nanotechnology to observe catalysis by individual enzymes and substrate molecules by fluorescence microscopy. Fluorescence microscopy can now detect it, which is the advance of the nanotechnology or the nanomedicine. Even the rate of single catalytic event and processes, individual steps in catalysis is measured by this process. So, these are the advanced process you should know what we have discussed after the inhibition part. Understood the thing? This is the thing. एक मिनट वीडियो एक प्ले कर देते हैं. Next class will be isoenzyme. Monday I will take. Monday your exams you don't want to give, no? You will be postponing. I don't know about the exams. If exam is not there, I have to.
Millions of biochemical processes necessary for keeping us alive. The pathways used to convert substrates such as glucose into products are
If
collectively
Monday exam is not
referred
there, I
to
will take the
as
class.
metabolic pathways. The drivers of these metabolic pathways are enzymes. Enzymes are usually proteins that work to assist chemical reactions by building or breaking down molecules. However, enzymes don't drive reactions at a constant rate. The rate of reaction can speed up, slow down, or stop completely
This is the
according
one-way
to the
traffic I
cell's
am speaking
needs.
about.
This process is analogous to an assembly line where raw materials are converted into products. The assembly line must be monitored to ensure that the supply of products does not exceed demand. If products are being created at a rate that is faster than they can be used,
This
Both
happens when you have
resources
enzyme
and products are
more
being
products which is
wasted.
not required.
The assembly line can produce at a slower rate or stop completely to prevent waste. In cells, this process is called feedback inhibition. Feedback inhibition is achieved through a process called allosteric regulation and plays an important role in many metabolic pathways. Enzymes have a unique area called the active site where substrates can bind. Enzymes that can be regulated have an additional area called the allosteric site. Downstream products can bind to this regulatory site, causing the enzyme to
This is a
change
simple
shape and prevent
example
it from
how to
binding
understand
with its
pictorially.
substrates.
Try to understand.
This causes the entire pathway to halt, and new products are no longer formed. Conversely, when the amount of downstream product gets too low, the enzymes interact with their usual substrates and begin forming products again. Feedback inhibition plays a critical role in preventing cells from wasting resources and energy. Mutated enzymes that do not respond to allosteric regulation have been linked to disease states such as cancer. Many processes in our bodies rely on feedback inhibition to keep everything running smoothly and efficiently, maintaining homeostasis.
So, thank you, and next class we will meet what we will be going with the last part, that is clinical enzymology. Last part of the enzymes, that will be the clinical enzymology and its different actions and its utility. Thank you.